Hi there, I'm Paul. This is Science That Sticks. Today, diffusion, osmosis, active transport and the required practical for biology paper 1. Roll the graphic. Hi there, welcome back. This is Science That Sticks. Once again, today I'm going to go through the three main factors that allow you to really answer the required practical that involves osmosis and potato chips and a variety of other things. So the first thing that I'm going to talk about is diffusion. Diffusion is the movement of particles from a high area of concentration to a low area of concentration. This can simply mean gas particles in a room. This could mean particles that are of a soluble solid that have been placed into a solvent to make a solution, and without stirring them, they will diffuse through the liquid. Osmosis is the movement of water into or out of a place across a partially permeable membrane. When I was a kid, it used to be semi-permeable membrane, but now it's partially permeable membrane, although I'm told you'll get the mark for either or. And this is again to do with from high concentration to low concentration, but osmosis is only about water, only water. So bear that in mind. Again, it's across a partially permeable membrane. In this instance, we're talking about the cell membrane. Now, active transport is a very similar kind of thing to osmosis and to diffusion in that particles are moving. However, it is different in the following ways. One, the particles are moving from an area of low concentration into a high concentration. That means it's against the concentration gradient. You must state that. Also, generally speaking, it's across a membrane because we're normally talking about it going into a cell, or rather into a particular tissue. This is true, for example, in the root hair cell, and not only is it true in the root hair cell, but it's also true in some of the microvilli in the intestines as well. Now, when you're asked a question that's going to require active transport as an answer, really think to yourself like, right, is there a picture? If there is a picture, is there a mitochondria present, and has it been labelled as mitochondria? Now, mitochondria releases energy from respiration. Now, that's the glucose plus oxygen that gives us carbon dioxide and water vapour, or rather water, which of course diffuses out of the cell and into the bloodstream, and then we respire and so on. Now, those key ideas are going to be useful for the required practicals. Now, the required practical normally talks about potatoes, and the potatoes are cut into chips that are the same length, the same surface area, so you normally measure them out and you cut them into pieces. Now, most of the time it's potatoes, but sometimes they might use any other root vegetable, so they might decide to ask questions about carrots or radish or swede. It doesn't matter what the vegetable is. What matters is the process that they are going to use. Now, you might be unlucky and you might be asked to write a method. If you are asked to write a method, then you simply do it as follows. Make sure that you cut 4 or 5 equal strips of potato, root vegetable, whatever. Make sure they are the same size, the same length, width and depth, so they are the same volume. You dry them first. You get 4 beakers or 5 beakers, depending on how many pieces of chips that you're going to go through. You make sure that all the pieces are dry, and then you weigh them. They have to be dry when they're weighed so that the water that's present doesn't upset the mass values. Then into the beakers, you have different concentrations of sugar, but in one of them, there's just water. No sugar is placed into it. The next one, you just have some arbitrary value concentrations. I've got 0.1, and then to the side of that, I've got 0.5 and then 1.0 grams per decimeter cubed. I've used grams per decimeter cubed because it's a biology question, so they don't care about moles. They really don't. And also it opens it up to both foundation and triple students. So it's kind of nice. Then what do? Well, you place all of the chips in the solutions for the same amount of time, say an hour. Then after an hour or a day or whatever it is that you want, you bring them back out of their solutions, you dry them, you reweigh them, and you measure the mass differences. Once you've done the mass difference, you are then in a position to then identify the change and percentage change. It's very important that you know how to do the percentage change. It's a little bit of maths, and because your biology paper is 20% maths, I guarantee that they're going to ask this sort of question or a question like it based on results. So that's the required practical, how to do it. Let's look through the analysis of the data, how to do a change, okay? Now then, for this particular example, I've made this data up. I don't know if it's accurate data or not, and I don't really care. What I care about is the process of you being able to do this. You will be given data in an exam situation. If you're doing it in real life, then obviously you would generate your own data. But because this is an example, I've taken chip 3. So chip number 3 started off at 2.05 grams. It lost mass. It went down to 1.95 grams. That's a change of 0.10 grams. To do the percentage change, you need the change divided by what you started with at the beginning, and then you times it by 100. So in this case, the change happens to be 0.10. The start mass was 2.05. You divide one by the other, multiply it by 100, and that gives you 4.878, and it carries on a bit. Now, if you've got an answer like that, the question might ask you to express it in terms of significant figures, maybe one significant figure or two significant figures. Now, that being the case, you obey what it tells you to do, but pay attention to that particular comment that they've made to so many significant figures. Remember, the first significant figure is the first number that you come to in any line. If it's a 0, so it's 0.000, whatever the number is that's not a 0, that's your first significant figure. Everything else, so if it's 1.2579, then number 1 is the first significant figure. Okay? That should be it. That should be all you need for this. Take good care of yourselves. Till next time, stay cool. Goodbye.